Hi, everybody, and thank you for coming to this. This next session, linking metal biogeochemical cycling from the atomic to the landscape scale, is a great segue from what has just been talked about. Although as not the intended moderator, I was very happy to be asked to step in because this topic, in my opinion, is one of mineralogy's most fascinating and societally important subfields. So when we are asked, what good is mineralogy in the 21st century by non-scientists who think that being a mineralogist just involves looking at hand samples of quartz, or other people in our own departments, hydrogeologists and geophysicists, I'm going to pick on them, who mainly remember looking for 2Vs under the optical microscope when they were undergrads. And one of the things that we can say is that the following two talks will allow us to best convince the public that nano-mineralogy is a critical component of our lives. So, first, I would like to introduce David Singer. He's from Kent State University, from atoms to mountains, new frontiers in X-ray science, and after that, Michael Schindler from Laurentian University, who will be talking about nano-mineralogy, a new dimension to understand the fate of metalloids in the environment. And again, we will not have questions after the first talk. We'll save it for the discussion. Thank you.
All right, good morning, everyone. Thank you for coming. And thank you to the organizers for giving me the opportunity to speak today. I'm very pleased to be here. This is an amazing space, an amazing building. This is not quite atoms to mountains, but does give you a sense of the scope of the scale of things that I'm going to talk about today, of colloids or nanoparticles or approaching nanoparticles of pyrite coated by iron oxides, soil aggregates that are mixtures of coal mine waste and soil, and then some things that are at the hill or at least maybe tiny mountain scale. And I'm going to come back to these pictures a little bit later on. I want to start with just thanking the mentors and supervisors I've had along the way who helped give me a good footing to start out my faculty position and help me get me to where I am today. Starting with Mark Furman, who I did a Department of Energy undergraduate fellowship with at Brookhaven National Lab. Mark's now at the NRC, Nuclear Regulatory Commission here in DC. And Mark gave me the first opportunity to apply mineralogy and geochemistry to environmental problems. And that first step, a little over 20 years ago, is not so far from where I am today, amazingly enough. Gordon may remember this adventure with Francois Farge to Trail, Mexico, where among other things we went to the Nika gypsum mine. I'm grateful for Gordon for the opportunities that I had during my PhD. And Glenn Wychunas and Jim Davis, who I did postdocs with. This is Glenn and I at the Advanced Photon Source outside of Chicago doing the exact opposite of what you're supposed to do during a tornado warning. Instead of seeking shelter, we, like everybody else, wanted to go see the storm. So that's what we were doing. As synchrotron X-ray sources were developed in the 1970s, it became apparent by the end of the 70s, early 80s, that these sources could be applied to a wide range within Earth sciences. I won't say quite how old I am or quite how old Gordon was when all of this was starting, but I would note that by the mid-80s, by the late 80s, it became pretty obvious that from an environmental mineralogy, geochemistry perspective, these could be pretty powerful sources. This was part of the first study that Kim Hayes published at Stanford in 1987 in Science that explored the mineral-water interface and how selenium, as selenium-6 and selenium-4, interact with the surface of goethite. Where using XFs, they could say something about if selenium was either outer sphere or inner sphere and actually say something about the coordination environment in a way to get direct chemical information at that interface, whereas prior to this time, we could only get that through indirect proxies. Moving forward into the late 90s, it became clear that these techniques could provide molecular scale information at the mineral water interface and the complexities that occur as a function of the geochemistry and biogeochemistry occurring there, especially relevant to processes within the rhizosphere. So Gordon and his students published this figure in Science in 1999, just prior to when I showed up for graduate school. And when I got into the group, this was the new paradigm. This was the starting point, at least for me, to think about, okay, these are the processes that are relevant. Where do I fit into this, and where do we as a community start to explore and try to figure out how all of this stuff works? And by the early 2000s, a couple of parallel approaches came to moving the field forward. The first focused on developing cutting-edge techniques, including synchrotron techniques, to really get at deeper and deeper molecular scale information to really highlight some of the important processes happening in here. And the other, which I'm going to focus on today, was trying to understand the fact that the processes happening within this schematic here, within this figure, are occurring there's a difference of 6 to 9 orders of magnitude in terms of what's actually happening in soils or sediments, and trying to design experiments that cut across this scale and put it in the larger hydrogeochemical and hydrobiogeochemical context in which they're forming. And that's what I'm going to focus on today. A quick note about the critical zone, this thin veneer of the Earth's crust that supports life all the way down to the bedrock and then up to the topsoil and the air above it. Within the critical zone, this is an incredibly physically and chemically complex part of the Earth, including coupled processes that can be seen across scale, down from the grain scale and subgrain scale, so at the millimeter down to the micrometer and submicrometer scale, up to soil aggregate or millimeter to centimeter scale, the pedon scale at, say, meter scale, and then larger regional scale as well. And we can think about the critical zone as being this patchwork of really diverse microscale environments that have a wide variation in both geochemical and biogeochemical processes. And the summation of all of these processes has a strong degree of control of the movement of material, particularly solutes, through the critical zone, whether it's contaminants or nutrients. I'm going to focus mostly on contaminants today, but we could have an equally fruitful discussion about nutrients. And nutrients that verge into contaminants, things like phosphates or nitrates. And I'm going to focus mostly on synchrotron techniques and how they've been integrated into both laboratory and field experiments that try to put that hydrogeochemical, hydrobiogeochemical context coupled with mineralogical data to really try to give us a full picture of what's happening here. I'm going to start first at the micrometer down to the sub-micrometer scale, and then start moving up into the soil aggregate, and then into the pedon and even landscape scale. At the smallest scale, at the micrometer down to even the nanometer scale, I just want to touch briefly on mineral surface coatings, where some of the really fascinating mineralogy and biogeochemistry can happen within the critical zone. I'm not going to say too much about this because I think Michael's going to speak a little bit more about it in the next talk. But these natural mineral coatings, these surface coatings, play an important role in controlling both nutrient and contaminant transport in soils and sediments. They can often have very complex mineralogies and a complex interplay of mineral phases, of inorganic phases, and organic matter. And the structure and composition are dependent upon the underlying composition of the grains on which they sit, and then the hydrogeochemistry, the hydrobiogeochemistry happening around it, all of which can result in physically and chemically complex systems. And some key features of them are the formation of intergrain fractures where there could be internal surface area, secondary coatings on top, and then also discrete aggregates. Unknowingly, I got myself into this world as part of my PhD project. I had the opportunity to work on uranium and copper transport within the Hanford 300 area. These were contaminated surface sediments of cladding and other nuclear materials where uranium and copper moved through the subsurface through preferential flow paths. These are reflected, visible light images on the top, and then uranium X-ray microprobe fluorescent maps on the bottom showing the distribution of uranium within these sediments. And we found evidence for uranium primarily associated with clay coatings of secondary clays, as well as the formation of discrete aggregates of silicate and phosphate phases, in a way that you would not necessarily have predicted based on bulk geochemical and bulk mineralogical thermodynamic data. Again, I'm not going to get into this too deeply. I'll leave this a little bit for Michael, and we can come back to this in the discussion. But we have, as Mike just showed, a very high degree of ability to characterize these nano phases, these sub-micrometer phases. And this is a focused ion beam TEM section showing that interface between the primary grain, in this case quartz, and then the secondary mineral surface coating above it, which is a complex mixture of mineral phases and nanomineral phases. And we have, through electron microscopy, the ability to define this physically and chemically complex environment, but trying to understand the fate of, say, nutrients or contaminants within these nano regions, or at least certainly sub-micrometer regions, is a little bit more tricky. One thing that might help is the development over the last few years of X-ray nanoprobes that can be run in scanning mode that don't necessarily get to atomic or true nanometer scale resolution, but do open up the possibility of tens or hundreds of nanometers of spatially located chemical information, where we could say something about, well, if there's arsenic or uranium moving through these materials, to actually get down to that sub-micrometer scale and say something about speciation and distribution. As we start to move up into the soil aggregate, so moving to the soil aggregate regime, moving from the nanometer and micrometer regime to the micrometer up to the millimeter and possibly centimeter scale, I'm going to talk just briefly about what soil aggregates are. There's not one type of soil aggregate. They're usually a mixture of materials, including naturally occurring organic matter, primary silicates and aluminosilicates that can often be porous. There can be an organic component, including living organic material like microbes or fungi, and then a whole range of secondary minerals, whether they're silicates, aluminosilicates, carbonates, or oxides, all in a complex setting. And that's ultimately where the composition and structure is ultimately defined by the native lithology, climate, landscape, and this interface between the geochemistry and the biogeochemistry. We've developed some pretty neat techniques to be able to describe both the physical and chemical environment of these materials, including some non-destructive 3D techniques to allow us to get at that structure and composition. So this is synchrotron-based X-ray microcommuted tomography. So these are soil aggregates. These are a few micrometers on a side, where without having to pull apart the soil aggregate, they could put these pieces of soil aggregates into the path of the X-ray beam and do the tomography and be able to say something about the internal structure and the mineral composition, particularly focused on iron oxides and clay minerals within the structure and saying something about their spatial distribution. And in this case, also focusing on the transport of a potential contaminant, in this case cesium or radioactive cesium, and seeing preferential flow paths and how the cesium is interacting with minerals within this material. Another example of microtomography being applied to soils, these are soils that we're developing on bauxite mine residues that are weathering over various amounts of time. Here, 1, 10, or 20 years worth of weathering, and the soil could be put into the path of the beam intact without having to pull it apart, without having to do substantial processing in order to say something about the structure of the material as weathering is occurring. As soil forming processes occur on these bauxite residues, there were significant increases in surface area, changes in porosity and porosity pathways, and the shape of the pore spaces between them, and saying something about how the aggregates are stabilizing or not over time. We have the ability also to do X-ray fluorescence microtomography and add in chemical information. In this case, this is a cross-section through a plant seed without having to physically cut through the seed. This is a virtual X-ray cross-section through the seed. In this case, this was a study that was focusing on manganese uptake through the plant from seed to sprout and then into a plant, trying to understand genetic and DNA controls over how manganese is removed and move through the plant, but this color map shows how manganese is specifically localized within parts of the seed in contrast to iron and zinc, which are in other places. This has the ability to open up trying to understand metal transport at, say, plant root mineral interfaces or micro mineral interfaces without having to do processing where we were potentially losing or damaging information along the way. In addition to getting at this spatial scale, it's also important to think about how we might improve our temporal information as well. And I want to touch briefly on using quick scanning X-ray absorption spectroscopy to get at sub-second geochemical and biogeochemical information. So using fast motors and improved fluorescence detectors, we can start to get at redox reactions or dissolution precipitation reactions that are approaching real-time information. So this was a study that looked at arsenic-3 oxidation by hydrous manganese oxides, showing data that could be obtained from the quick XAS in contrast to batch experiments, and getting into the sub-second timescale of the reaction in a way that in traditional bulk experiments we could not get to that. This was a study that looked at iron 2 catalyzed transformation of ferrihydrite and getting full XF scans out to K of 15 inverse angstroms in 20 seconds. So getting a full XF scan in 20 seconds. And this is for a bulk material where you have lots of iron. We could have a discussion about how you would do this for trace elements, which are not going to get full XF scans in 20 seconds per se. But being able to track the change in the mineralogy of this material, again, at the sub-second time scale, and being able to say what's happening in the mineralogy of the system. And again, we're starting to approach our ability to get at the kinetics of geochemical and biogeochemical reactions, whether they're redox reactions or dissolution precipitation reactions. I'm going to start now moving up into first laboratory and then field-based experiments as we're increasing our scale of what we're looking at. First, looking at soil aggregates, of synthetic soil aggregates, to try to get at some of the interesting things that are happening within actual soils. These were a series of studies done by Celine Palud as part of her postdoc at Stanford, looking at artificial centimeter scale soil aggregates. These are golf ball size ferrihydrite coated sand, which was then inoculated with iron 2 and iron reducing, that were inoculated with iron reducing bacteria in a food source, hence the lactate. And this shows a cross section of the, lost her. Turn that on. Oh, there it is. Okay. Showing a cross-section through the soil aggregate, and then after 30 days, clearly showing a change in the mineralogy of this material. And these were studies that were done to try to mimic what's actually happening in natural soil aggregates with fewer variables in order to try to do that. They could track over time and over space the mineralogy of the iron-bearing phase, showing changes from the interior to the exterior with the corresponding color change. And they could say something about what is happening to the mineralogy as a function of time and space as iron is diffusing from the inside out, resulting in secondary precipitation of goethite and lepidocrosite. Over time, though, what's happening in the interior is starting to change as there is a buildup of reactive species, including the potential for the precipitation of siderite, but a greater degree of goethite and lepidocrosite. These studies helped illustrate the importance of microzones within these soil aggregates, and that the variation within these microzones can truly happen at the micrometer scale and changes from advective to diffusive flow at that micrometer scale. And these soil aggregates these artificial soil aggregates are important steps towards trying to mimic or at least model what's happening in naturally occurring settings. As an extension of this work, we could then think about what happens if there's a nutrient or contaminant moving within this system. So these are the same golf ball size soil aggregates of ferrihydrite coated sand, but now they've added arsenic into this. So these are X-ray fluorescent maps, X-ray microprobe maps of iron and silicon on top. Clearly you could see the iron coated the quartz, and then adding arsenic 5 into the system and tracking over time and space the production of arsenic 3 at the expense of arsenic 5. And you can see over time, now the cross-section through this, there is still this oxidized rim and then a more reduced zone in the interior, using microzanes to track the production of arsenic 3 and then the loss of arsenic 5 across time and across space. These oxidized rinds that are on the exterior of these artificial aggregates serve as a barrier for arsenic and also iron 2 to diffuse out, but do result in the buildup of arsenic within these regions. And changing geochemical conditions could easily result in a pulse of these materials out, again highlighting the importance of microzones within this. As we start to scale out to the pedon and the landscape scale, I want to focus on one study that was done in northwestern France that focused on some of these complexities within riverbanks. And riverbanks can be considered to be a biogeochemical filter between upland wetlands that are sourced to them and then the rivers that they're connected to. This was a study that was using bulk XFs and a number of other techniques to track the mineralogy of iron within the riverbank that had a fluctuating water table. And so the surface was primarily oxidized but periodically anoxic, and then a subsurface that was, for the most part, anoxic, but periodically oxidized as well. And as the water table fluctuated, they were able to track changes in mineralogy, both as a function of depth, so the more shallow versus the deeper particles, and then as a function of particle size as well, and could say something about how the iron was distributed. For the most part, in the near surface, iron was dominated by nanoparticles of ferrihydrite and nanophases of iron mixed with organic matter. And then in the subsurface, due to subsequent changeover, there were more transformations that were occurring, resulting in a buildup of goethite. As those changes are occurring, trace elements, particularly contaminants that are moving through the wetlands into this system, are starting to be accumulated, but in ways that are controlled by the mineralogy. So, for example, the iron oxides would accumulate arsenic, the organic matter accumulated lead, and then cobalt, copper, and nickel could be accumulated by these iron organic matter complexes. What's unique about this system is that the buildup of the iron oxides and then the trace metals that are associated with them at these riverbanks could then be periodically flushed through weathering of these riverbanks. And so seasonally, we see that they observe changes in how the trace metals were built up here, and then a seasonal release of a pulse of colloids, of colloidal-bearing material, where these trace metals could be released. And so it's not just a slow, steady bleed of trace metals out of these wetlands into this river. There was this seasonal variation of these colloids being transported out of this system. Moving up even to the landscape scale, to the kilometer and beyond scale, this is a study that looked at soil weathering as an engine for manganese contamination in groundwater, focusing in the Piedmont region of North Carolina and more broadly in the southeast. Manganese is a recognized health concern as a secondary EPA contaminant, and it's been linked to a whole host of health issues in humans if you drink too much, especially if you're drinking groundwater that's not properly filtered. And this study used a combination of field, laboratory, X-ray spectroscopic, and geospatial analyses to show how manganese is moving from the fractured bedrock into the groundwater zone, showing that weathering of the bedrock cycles manganese between primary minerals and secondary manganese oxides, and in that process resulting in the release of manganese into the groundwater. And being able to make that link from what's happening at a small scale out to a larger scale. And it's estimated that over 1 million people in the Piedmont region are drinking groundwater that has excess manganese in it, which represents a long-term risk. I want to touch on, so I just mentioned colloids. I just want to talk a little bit about colloids, and I'm not going to get into the discussion of what's the difference between a colloid and a nanoparticle. We can come back to that as part of the discussion. But colloids, or moving material in these tiny materials, can be thought of as biogeochemical scale cutters. They cut across scale. Characterizing them and studying them requires working at the micrometer and submicrometer scale, but they can move material across the centimeter or meter scale in soils and over the kilometer scale if they're moved into a stream. And there are some really unique ways in which they interact with organic matter, both living and remnant organic matter in soils or in sediments, in ways that drastically can change the potential bioavailability of whatever metals are associated with those colloids. We also can discuss what's the best way to sample for a colloid if it's in a stream versus if it's a soil or sediment, the vadosome versus the groundwater. What is the right way to sample for colloids? We can get into that in this part of the discussion. But we can see just through one example of how colloids can be vectors for contaminant transport. This was coming back to arsenic and coming back to iron oxides. This is a study in Spain that used asymmetric flow, field flow analyses with ICP-MS. And so they can take the stream of particles and separate them out by particle size, send that through the ICP, and then determine where, in this case, arsenic or other trace metals are found as a function of size fraction. And they could see that the arsenic was primarily associated with these nanometer scale particles that were described by TEM with EDS and then by XFs as iron oxides, but a mixture of iron oxides and organic matter that are transporting arsenic out of these systems, and often long distances from, and at the kilometer scale, long distances from where they're forming. I just want to touch briefly on work that I've been part of with my colleagues and our students. This is in the northwest corner of Appalachia in Ohio, of an area impacted by historic coal mining. This is a tributary from a heavily impacted watershed that's been upstream, has been reclamated. This is a beaver dam, And then below the beaver dam, you can see this orange color of the water. Clearly that water is not remediated. There's likely a groundwater source of acid mine drainage. Sampling colloids and suspended particles in here, we can see these large aggregates that are a mixture of, you can see this really beautiful framboidal pyrite, and a mixture of iron oxides, organic matter, silicates, and alumina, and secondary minerals as well. And this is pyrite surrounded by iron oxides, indicating just how in disequilibrium these particles are that are transporting materials over a long distance. And we can use X-ray fluorescence mapping and doing redox mapping to map out sulfur in terms of its in different oxidation states, saying something about the iron redox state distribution, including the formation of oxidized rim on these particles, and linking that to the broader mineralogy. And so I'm going to leave these pictures back here. Again, not quite from atoms to mountains, but we have looked at a wide range of scales. And in terms of the outlook for where this is going, I think we are pretty good at coming up with really novel ways of laboratory and field experiments to try to understand this link in scale. But I will leave more for that for the discussion. So thank you very much for your attention. I appreciate it.